Hi, good afternoon, everyone. Welcome to another section of your meeting. And this afternoon, I'm very delighted to introduce my colleague, Professor Aline Petrin, from the University of Iowa, United States. Professor Aline, she's Brazilian, by the way. She went to University of Iowa a long time ago as a doctoral fellowship. She developed a lot of work there, and she became a professor at the Department of Orthodontics in the University of Iowa. She's also an associate research scientist at the College of Dentistry in Iowa University. So she's an inspiration for everyone who wants to follow this path. Her current research is focused on twin approach for genome-wide differential DNA methylation in orofacial cleftings and genetic and genetic interactions underlying the challenge of orofacial cleftings. So Aline, thank you very much for accepting this invitation and feel free to give your lecture.
Thank you. Do you all hear me okay? See the slides? Is it good? All
Yes.
right.
Okay.
So first, I want to thank the organization of this meeting to give me the opportunity of presenting for you today. Like Professor Lucilene said, my name is Aline Petrin. I'm an assistant professor at the University of Iowa College of Dentistry. And today I want to present you a little bit about genetics and epigenetics of cleft lip and palate and how do we use genetic and epigenetic approaches to study and discover genes that cause this condition. So I'll go over a few examples and methods and I'll show you my own work and work of my collaborations as examples of what can we achieve when we use these methods. So first, my main focus today will be non-syndromic clefts, but I also want to tell you that clefts can be part of syndromes, and there are over 300 syndromes that have cleft lip and palate as part of the phenotype. And the most common syndromic form of clefting is the van der Waals syndrome, which is an autosomal dominant syndrome and affects about 1 in 33,000 births. And the phenotype may include cleft lip, cleft palate, and hypodontia, and also lower lip pits. But van der Waals syndrome is not a complete penetrance, and because it's not completely penetrant, sometimes patients do not have the lipids, so they might be confused in a clinical examination with non-syndromic clefting. But because van der Waals is an autosomal dominant, these patients will have 50% of chance to passing the syndrome to their children. So it's really important to know the mutations that cause and screen these patients for mutations so we can give them a proper genetic counseling. We know that mutations in IRF6 and GRH03 cause van der Waals syndrome. And in here on this side of the slide, I'm showing you the first monozygotic twins, the identical twins that were sequenced here at the University of Iowa, and that allowed us to discover that IRF6 was the causal gene. Then after that, we conduct expression analysis showing that IRF6 was highly expressed in the medial palate and tooth buds and hair follicles and also skin. One interesting fact about IRF6 is that coding mutations cause the syndrome, the syndromic form of clefting, and the non-coding mutations in the same gene contribute to non-syndromic cleft lip and palate. So it's a perfect example of how the study of syndromes can be a surrogate to discover etiological factors for non-syndromic cleft lip and palate. And non-syndromic cleft lip and palate are much more common than syndromic forms. And that's the reason why we need to explore all different methods because there are over 50 loci that have been identified so far that contribute to non-syndromic orofacial clefts. So they're the most common craniofacial birth defects. The incidence varies according to the ethnicity, with Asians having the highest frequency and Africans having the lowest. 70% of cases of cleft lip and palate and about 50% of cleft palate only are non-syndromics. And usually in etiological studies, we divide them in cleft lip and cleft palate only because there is evidence of distinct etiologies for these three types. But we also know that clefting is not, there is also an environmental component to it. And we've been learning a lot about this interaction between genetics and environmental via studies of the epigenetics factors. Some of the environmental components of clefting etiology can be smoking, alcohol, nutrition, teratogens, obesity, and diabetes. All of this has been shown to be associated with clefting. And we have hypotheses that specific alleles in metabolic pathways increase the risk for orofacial clefting, especially with smoking, alcohol, and nutritional deficiencies. But not only the maternal genotype must be studied, but also the fetal genotype. It all depends on how the maternal and the fetal genomes interact. And I'll show you a little bit. It will make a little more sense in the next slides. So let's use alcohol as an example. In a very simple way, after alcohol is ingested, two main enzymes, ADH and ALDH, help break apart the alcohol molecule. And first, the alcohol will be metabolized into acetaldehyde, which is a highly toxic substance for the organism. And then in a second step, acetaldehyde will be metabolized into acetate, which is then broken down into water and carbon dioxide for easy elimination. But let's say we have a mutation that affect this conversion here, and it slows down the metabolism between alcohol and acetaldehyde. That will cause a higher concentration of alcohol in the blood. We can also have a mutation that accelerates this process, which will cause an excess of acetaldehyde, which is makes people more sensitive to drinking because those people that can only have a little bit of alcohol and just feel a terrible headache, a terrible hangover. So that could be due to mutations in this part of the pathway. On the other hand, mutations that will slow down this step will also cause an excess of acetaldehyde. Or if it accelerates, it makes people a little more tolerant to drinking. So we can see from here that the genetic variations affect the way people metabolize and tolerate, in this case, alcohol. We can also think about it using smoking. In the effect, the maternal smoking may be modified by genes involved in this biotransformation or toxic compounds derived from tobacco. So again, the toxins from smoking, they are broken down in a very simple way into intermediate metabolites and then in water soluble for excretion. And mutations that are changing either phase one or phase two of of this metabolism will also change the way that person metabolites and control. So one of the studies we have here, we are looking into the interaction between the maternal and the child genetic variation in genes of the smoking detoxification pathway and how that affects the risk for clefting. And we do see that both genomes are important, not only the maternal, but also the way the child is responding to that exposure. I created this timeline just to give you an idea of the most impactful genome-wide studies that have been done in the past few years. And as you can imagine, there's a lot of money, time, and resources invested in here. And here in this top part, I have the genetic studies. and down here I have the epigenetic and environmental studies. Most of the early part of the 2000 years were focused on GWAS and copy number variation studies, and also interaction between gene and environment. And then we started seeing a more extensive studies including whole genome sequencing, and now the epigenetic studies are gaining a lot of ground in the clefting field. So let me tell you a little bit about genome-wide study association studies, and they are most used for identification of common variants in common complex traits like clefting. There are the mutations that are rare, and then they have a very high impact, and when a person has one mutation it's enough to create a phenotype. But like in the case of non-syndromic clefting, it's usually the combination of many lower impact variations that increase the risk for the phenotype. And that's not only with clefting, it's with many other complex diseases. Because the effect is so small, we need thousands of cases and controls for a efficient genome-wide study, and we test millions of SNPs per person and generating billions of data points. So as you can see here, let's use these two SNPs in chromosome 9 as example, and we test thousands of cases versus thousands of controls, and we can see that the frequency of these SNPs is much higher in cases than in controls. The same for SNP2. And when we plot all the millions of SNPs together, we come up with a Manhattan plot, which will show us exactly the ones that are more frequent in cases than in controls, and it will generate a genome-wide significant p-value. And that's how we end up identifying common variants, which means it's present in many people, but it's more frequent in people who have the phenotype that we are studying. Okay, so in this case, I'm just showing you Manhattan plots from studies we did here in Iowa and with our collaborators, and in which we discovered hits for cleft lip and palate, for cleft palate only, and for all kind of clefts combined. And you can see here that when we see all clefts combined, the signal that we had for cleft palate only disappeared. So that's telling us that this gene is specific for cleft palate only, and it's not as relevant for cases with cleft lip and palate. So there's a lot of information that can be drawn from genome-wide studies like this. Another thing we learned from GWAS is that the associated SNP is not necessarily the one that is the theological variant, because there is something we call linkage disequilibrium. Some of our SNPs, they travel together in like intervals, like a little recombination, and we call those linkage disequilibrium blocks. And in addition to linkage disequilibrium blocks, because our chromosomes, they're not linear structures, right? The DNA is, there is a 3D conformation. We end up generating what we call topological associated domains, which are regions that the DNA physically interact more often than with other more distant regions due to this conformation. So, using this information, we studied a series of regions that have cleft associated SNPs, and we came up with cleft associated topological associated domains. This allows for additional studies, not only the pinpoint SNPs that we were used to when we were doing only GWAS, but that also allow us to look for different etiological factors, for example, regulatory regions that will be interacting with the SNP of interest, and also for epigenetic changes, which is the focus of my research. Let me shift gear here and talk a little bit about copy number variation. So moving up in that timeline, we had a first block of GWAS studies, then we move on to copy number variation. And it's been shown that the human genome differs a lot more at a structural level than at the nucleotide level because it makes sense. Like a deletion, it will take a big chunk of the DNA, and it might have a much higher effect than just one single base pair that would be a SNP. And approximately 12% of the human genome is copy number variable, right? And because of it, the size of these deletions and amplifications, it involves at least 3 times more nucleotides than single SNPs would. And one of the best way to detect copy number variation in the human genome is through doing array-based genomic hybridization, which is an array that we hybridize the DNA of the patients and we have intensity signal based on the number of copies. And using this mechanism, we hybridize samples from 97 cleft lip and palate cases and 43 cleft palate only, and we discovered a new candidate gene for clefting. And it was a larger, we had 2 patients with this deletion, one a very large deletion, which didn't really tell us which gene it was because there were 2 genes included there. But then we had a second patient that narrowed down our region of interest to this gene ismin-1. And after that, we studied in frogs, and we were able to see that the ismin-1 was expressed in the midline in the mouth, around the mouth of this frog, and the one that was a knockout had a midline cleft. So that gave us an additional piece of information towards ismin-1 being a new candidate gene. Now, the new studies are much more complex and larger, and they're doing whole genome sequencing, which is a high resolution base by base view of the genome. It captures both large and small variants, and it has the potential to identify causative variants, both common variants and rare variants, and it generates a much larger amount of data. And with whole genome sequencing, we here in Iowa, with our collaborators in Pittsburgh and other universities, we were part of the Gabriella Miller Kids First pediatric research program, and we have whole genome sequencing for over 900 case parent trios. And we found common variants for 315 European trios and 265 Latino trios. And we identified a region on chromosome 21, which seems to be specific for the Colombian population, and that is the associated SNP. And that also led to a new region of a new cleft gene, and this SNP overlaps with a what we call a super enhancer, supercraniofacial enhancer. And I would give you much more detail if I could, but time permitting, I need to move on to the other part. But the craniofacial enhancers, the region that controls craniofacial development in the human in human embryos. So this is, like I said, it seems to be specific for the Latin population, especially Colombian population. So far I told you how different types of genetic variation can affect clefting, but there is another layer of information that controls gene expression and, if disturbed, can lead to a phenotype. And we call it epigenetics because we know our genes, they don't act alone, and there is a lot more going on than just genetic variation and the mutations and the polymorphisms. So let me begin by what is epigenetic. What do we understand by epigenetic? It's the study of heritable chains in gene expression that do not involve changes in the underlying DNA sequence. So in a simple manner is if you imagine that our genome is a textbook that is explaining us how to make the human body. Epigenetics is as if you get a marker and you start telling your genome, this part is more important. You need to express this part right now, but not the other one. So there are different types of epigenetic marks, and each one of them will tell us what part of our genome is important at that timing. And very importantly, epigenetic marks, they are reversible. So they can activate or deactivate part of the genomes in different timing depending on what timing of the development that the body is. And why is this important? Why is this relevant? So for decades, biologists way back in time, thankfully, biologists was always intrigued how one single fertilized egg with a single genome can generate the hundreds of different cell types that we have in an embryo or even in an adult body, right? Our genome is the same in all our cells. All our cells have the same genome. So how are they different? How do they turn out so different? And back, they actually thought that there was a tiny, fully formed human inside the sperm head, which was called homunculus, and that the egg, the mother, was really a vehicle to grow that tiny little human that came ready to go. Thankfully, now we know more. We know that that's not the case at all, right? And we know that what happened is that there is this additional information, there's this additional layer that regulates how the genome works, and that was called epigenetics. So the scientists use the term epigenetics to represent this extra layer of information that enable differentiation and development. This will let our body make different cell types. Although we all have the same genetic code, we are able to generate different cells at the right timing, at the right tissue. So in a way that our body works based on these distinct epigenetic marks and works properly, silencing or activating different genes at the time that they are needed. Epigenetics also contribute to tissue homogeneity, right? And it means that it tells daughter cell, daughter what they have to be. So one kind of cell will generate the same type of cell, making sure that the tissues are homogeneous and that they have a cell identity, which lacks in cancer. And that's when we see that tissue heterogeneity in the tumors, and we see different cell types in the same place, which is very common in tumors. Like I said, there are different types of epigenetic marks, and they all have their own role in activating or deactivating the genes and the transcription. In a way, we can say that they are switches. They can do on and off, but they can also be dimmers, telling the genome how much transcription has to happen, and it's all time sensitive and it's all tissue relevant. I will focus on DNA methylation because that's my area of research, and I have some interesting data to show you in the end. And DNA methylation is the addition of a methyl group to the DNA, to the cytosine, and we call those CPG sites. And it can the unmethylated gene will be transcribed. It's active, and when a CPG site close to a gene is methylated, it's silenced. So we know that not all our genes are active at the same time, and this is one form of regulating this activation and silencing. And there are different ways that DNA methylation can affect gene expression. Let's say an unmethylated CPG site will allow the transcription factor to bind in, while if it's methylated, won't be transcription. Their methylation there are methylation sites that there are transcription factors that are sensible to methylation, so they will not allow the binding, they will be blocked. There's also recruitment of different proteins that will sit on a methylated site and not let the transcription site sit there and start transcription. And there are also proteins that will recruit the transcription site and bring them to a site that is unmethylated and therefore start the transcription, like in these examples. So we know that DNA methylation and other epigenetic marks, they're responsive to environment, and our lifestyle can influence it, and it's been shown like in cancer, Alzheimer, depression, heart disease. There are many diseases that we know that the lifestyle contributes, but our lifestyles, it's way more likely to change our epigenetic machinery than our DNA sequence itself. But that doesn't mean that not changing the DNA will not cause disease, because as you can see, there's different ways that we can silence genes, even if they don't have a mutation. And the role of methylation is when the role of the DNA methylation is causal, we can study those marks and develop biomarkers, therefore helping to predict diseases. So I told you that epigenetics marks do not change the DNA sequence, but the DNA sequence can influence epigenetic marks. And here, I'm just going to give you a quick example. Let's say we have a binding site and our transcription factor here, and this site is not methylated. If there is a mutation in the transcription factor binding site and it turns methylated, the transcription factor will not bind anymore. Or if there is a mutation in which the in the transcription factor itself and it's not produced anymore, so there won't be transcription. Or if the methylation occurs in the binding site and then that transcription factor is not it's not sitting there and it's not compatible with the transcription site any longer, and you won't have transcription. So in these last few minutes, I'll give you an example of what my research is and how I use this epigenetic studies to apply for non-syndromic orofacial clefts. My study involves monozygotic twins, and like I told you before, epigenetic marks can control cell types, but what does it happen when we're talking about a whole human, like a fully grown human? Right? Those epigenetic marks, they are still working throughout our lives and making a difference. So here's a thought. We have two individuals that have the same DNA, and we call they're monozygotic twins. They have the same DNA sequence. It's plausible to think they would be identical in everything, but actually we know that's not true. About 30% of monozygotic twins, they are discordant for height, for example. They look alike, they're extremely alike, but they do have distinct traits. And what we've been finding more and more is that many cases of monozygotic twins will be discordant for a disease as well. In my case, I use discordant twins that are discordant for clefting to study their epigenetic differences and find out epigenetic risk factors for clefting. By using monozygotic twins, we can control for potential confounders because they're genetically identical. They have the same age, the same sex, they've been through the same pregnancy, and they've been exposed, at least during development when clefting is forming, is happening, they've been exposed to the same environmental factors because they were in the same pregnancy. So what I am interested in is comparing the epigenetic profile, the methylation profile between these twins that are discordants, to find what is different between them and is relevant to the one of them having a cleft, the other one not. So what I'll show you today is the results from 31 pairs of cleft discordant monozygotic twins. 20 of them had DNA from blood, 11 had DNA from saliva. We ran a very strict pipeline of cleaning and normalizing data. We ran the Illumina Epic array and interrogated over 150,000 methylation sites throughout the whole genome. And we correlated blood and saliva because I wanted to know how similar they are. And for that, I had blood and saliva from the same person, 4 people with with both tissues. So we did find a high correlation between blood and saliva, and that could tell us that in the future there's the possibility of even combining the both data sets, but we still need more information before doing that. We did separate them. We did perform analysis separately, DNA from blood and twins with DNA from blood and twins with DNA from saliva. And from that, we were able to find novel sites with differential methylation genes been in that have not been associated with clefting before. We did find differential methylation in genes that we know that are associated with clefting. We replicated loci from other cleft studies that were done previously to ours, and we found an overrepresentation of cleft related phenotypes when we ran a gene ontology analysis. And I just have just 3 more slides to show you the results very quickly. And in this table, I'm showing you the top 10 results for blood and the top 10 results for saliva. Like I said, we see genes that are hypermethylated and downregulated in situations that are relevant for cleft development, like regulators of bone homeostasis, opioid receptor, which would be related to the environmental exposures. There's a gene that has been proven to be involved in mice palatal development, and some genes that overlap with craniofacial superenhancers. And then in this slide, I'm showing you that when we ran the gene ontology analysis, we see over enrichment of terms that are relevant to clefting. For example, increased connectivity tissues, ectrodactyly, split hand, lip pit, like I showed you in the van der Voort. So this kind of analysis tell us that the biological results we are finding with the arrays are in the right path. We are finding things that are relevant to development, and we are finding that disruption of methylation in these genes is a plausible explanation for contributing to clefting etiology. Just to finish, I wanted to show you today the results from the first EWAS using monozygotic twins discordant for clefting. There is another study being done with monozygotic by another group as well. We show epigenetic marks that are essential for gene expression, that differential methylation is present in non-cleft genes and novel genes as well. And these studies, they help us prioritize region for functional validation and show us that methylation is a plausible mechanism for which environment can change gene expression. And it also gives us the possibility to use these epigenetic biomarkers and develop biomarkers to help prevent or even improve treatment outcomes in clefting. With that, I have a lot of people to thank. I'm very fortunate to be part of an amazing group of researchers. And above all, I thank the study participants. There's no human research without them. And I thank you for listening, and I'll be happy to answer any questions.
So thank you very much, Aline, for this brilliant presentation.
It's a lot of information for a short time.
It is, exactly.
I didn't confuse you too much.
Yes, but lots of information. I could listen to you the whole day long, but unfortunately we don't have just time. And actually, We don't have so many time for questions. So, and I have so many questions for you, but I'm going to ask you a question that is not related to epigenetics, because I'm very interested in copy number variation, and you cite a gene, the GRHL3 gene, and I'd like to know if you have found some copy number variation in this gene that could be related to orofacial clefting.
In GRHL3 or any other one? I don't have, in the cases I've done, I don't have any copy number variation in GRHL3. We do have SNPs in GRHL3 that contribute to non-syndromic clefting.
I'm asking that because you found actually was a micro duplication, but you thought that it's just like polymorph is not related to orofacial clefting. But when you mentioned that, it was like, oh, maybe something, you know. But it's a micro duplication.
Well, usually duplications are more tolerated than deletions, right? But it's worth remembering that GRHL3 mutations in GRHL3, they cause van der Vood. So even if the patient doesn't have a clefting, but it has any other abnormality that would be in that same pathway or gene network, that would be worth checking.
Okay, and also I'd like to ask you so many questions about the genetics, but let me see what people are asking.
Now you got my email.
So, yeah. So I ask him later. Okay, so Professor Katch is saying that she's interested in the reversible character of the genetics mutation. And okay, lots of people saying that it's amazing presentation. It was wonderful. Your explanation was very detailed. And congratulations. What else?
Thank you.
I'm sorry we don't have so many time. But,
yeah, I don't see. Oh, do I see? Oh, I do see. Okay, now I see them.
Okay.
Well, thank you.
Isabela, are you saying that something that I'm not saying? But I don't think so.
I'm sorry, Lucy.
Okay, anyway.
I can't hear you.
So, Aline, because we don't have so many time, okay, Professor Katch is asking if there is an observation of the reversion.
What do you mean by the reversion? Can she? I don't think if I.
She said that she's interested in reversible character of epigenetics mutations.
Okay. You mean the way that epigenetics can influence genetics or vice versa? Or
yeah,
I'm afraid I didn't understand her question.
She's saying that the epigenetics, I think, the way that epigenetics can reversible the mutation.
Oh, the reversible. Okay, yes. Well, there are cases where, for example, and I'm going to use alcohol or smoking. Smoking is a better one. So when people smoke for too long, they create genetic marks, epigenetic marks, right? But there is also evidence that when you cease that environmental exposure, the epigenetic mark is reversible. It will go back to normal. Let's say we smoke for 10 years and then you stop smoking and then it's reversible. It's possible to reverse. In our case, we're studying clefting, which is very early in development, so it's hard to say. And when in these patients, I got blood from them or saliva from them from later on in life. So that's one limitation when you're studying birth defects. And I see there's another, but yes, there is evidence of being reversible. And it's worth remembering that they will silence and activate genes in different times. and I see, is there a relationship between microRNAs and development? Yes, there is a lot of research involving microRNAs because microRNAs are a different way of controlling gene expression. We do have here in Iowa a project going on right now that we have blood from the child, we have tissue from the child, and we also have exposure information from pregnancy. And what we're doing is that we're doing the genome of this child, I'm doing the epigenome of this child, and then we're doing RNA sequencing of the same the tissue of this child, and we'll combine all this information to make like a big, huge omics, multi-omics study. And yes, there is a lot of microRNA studies going on for clefting. But again, the main limitation with clefting is the timing, because it's basically impossible to get tissue from a developing child at the right timing, right? That it's relevant for clefting. However, there is a big study, and I'm happy to share with Professor Lucilene the paper later on, that they did use human embryo, and they established epigenetic markers and different chromatin marks during the different timings of development, and that generated a database that tells. And when I mentioned craniofacial super enhancers, and that's how we identify the regions of the genome that control craniofacial development at the right time during the embryo development. So there is information, but it's limited because of the timing when clefting happened.
Okay, so we are out of time.
Yeah.
So thank you, Aline, very much
Thank you.
for your presentation and for accepting the invitation.
Thank you. Bye.
You're welcome.
Good afternoon, everyone. Once again, my name is Isabela Pavanelli. I'm co-hosting this section with Dr. Lucilene, and it's a pleasure to be here talking to you. Now I'm gonna introduce to you our next speaker, Dr. Robert Pogue. He is an adjunct professor at the postgraduate program in Genomic Sciences and Biotechnology at the Catholic University of Brasilia and visiting researcher at the Athlone Institute of Technology at Ireland. He has a background in human genetics with a primary interest in skeletal development. Professor Pogue is going to talk about molecular bases in genetic skeletal diseases. Thank you, Professor Pogue, for accepting our invitation and to participate in our event. Now it's up to you. Feel free to share your screen with us. You can start your presentation.
Okay, thank you very much, Isabela. And I would like to thank the organizers for inviting me to speak here at this event. It's always a pleasure. And so, without further ado, I'd like to present some of our work that we've been doing over the past few years regarding further investigations in genetic diseases of the skeleton. And so, what I'm going to focus on here is within the large group of diseases that we call genetic diseases of the skeleton. And if you look at the most recent nosology, which is a publication describing the different diseases, the categorization, and the genetic bases, there are at least 461 different known diseases that cause malformations and problems with growth of skeletal elements. For these, most of the genes have already been identified, especially in the last 10 to 15 years with the advent of next generation sequencing. And so far we have identified approximately 437 genes responsible for somewhere in the region of 425 of these disorders. In the current presentation, I'm going to tell you a little bit about work that we've done in relation to 3 different skeletal diseases and work that is ongoing. And then after that, I want to tell you a little bit about some of the functional in vitro studies that we have underway and some of the findings that we've produced so far. So the first condition I want to tell you about is called odontochondrodysplasia. And as the name suggests, this is a skeletal disorder which affects the skeletal elements and also the teeth. And so it's a congenital condition. The children are born with spinal changes, and they also have metaphyseal malformations. Also, as you can see, they are born with dentinogenesis imperfecta, which is a problem with the development and mineralization of the teeth. Now, at the time we began this study, which involved this little girl here, who was a child of consanguineous parents born here in Brazil, the gene was unknown. Now, due to what we might call an error in communication, as we were finalizing our work, another group in Europe published the identity of this gene, and we published shortly after. But what I want to tell you about is the path that we took to identify mutations in the TRP11 gene. Now, the TRP11 gene codes a protein in the Golgi apparatus, which is important for maturation and transport of proteins. When we did exome sequencing using the Illumina platform in a commercial laboratory, we found a homozygous mutation, as expected, due to the fact that this was a product of a consanguineous marriage. And the mutation was in the TRP11 gene, which is located on chromosome 14 and has approximately 21 exons in its longest splice isoform. The mutation that we found was not in the coding region, but was in a predicted splice site at the beginning of exon 9, so a splice donor site. Now, the interesting thing about finding mutation or a candidate mutation in this gene, and we could say the confusing part about it, was that this gene had already been associated with a much more severe skeletal dysplasia known as achondrogenesis 1A. And as you can see from these x-rays here, the children who are born with achondrogenesis type 1A have a much more severe phenotype. You can see that there are bone malformations. They have many short bones, including short ribs. And if these children survive to birth, they usually die within the first few hours or first few days after birth. And this x-ray is from another patient that we analyzed in the last couple of years. And this patient did survive to birth, but died within 30 minutes. So the question we had was, if the TRP11 gene is already associated with this very severe lethal skeletal dysplasia, how can it also be causative of a much milder phenotype? Obviously, the girl that I showed you in the previous slide is still living, and despite her skeletal problems, she is relatively healthy. Of course, there are many precedents in the literature about genes that can cause both severe and less severe phenotypes, but still, we wanted to try and explain what was going on. So doing a bit more analysis of this mutation, we went to look at the exon that would be predicted to be deleted. So when we looked at this variant at the beginning of exon of intron 9 of the TRP11 gene, we discovered that it would be predicted to delete exon 9. Now, looking at exon 9, we discovered that it has 87 base pairs. And if you do a quick calculation, you'll discover that 87 is a multiple of 3, which means that it causes an in-frame deletion. Right? So the prediction was, or initial hypothesis was, that we would get a shorter protein missing the amino acids encoded by exon 9, but that still most of the protein would be present and that it would still retain a good part of its function. And so this would help us to explain why mutation in the same gene could cause a lethal and a non-lethal phenotype. So going to the Ensemble website, we discovered that, in fact, there is already a naturally occurring splice isoform of TRP11, which is missing exon 9. So this is exon 9 here, present in the longest splice isoform. And there is another naturally occurring protein coding expressed isoform, which is missing exon 9. So this gave strength to our hypothesis that an isoform of the protein missing the amino acids encoded by exon 9 could still be at least partially functional. So we decided to get cells from the patient and see if these different splice variants were expressed. and to see if the analysis of expression through the RNA would support our hypothesis. So we isolated RNA from peripheral blood cells, and we synthesized cDNA, and then we designed primers to amplify from exon 8 to exon 11. And so we did the analysis here in some controls. Each lane on this electrophoresis gel that's marked with a C is a control. We also have the patient's mother, the patient's father, and the patient herself. So when we did our PCR amplifying exon 8 to exon 11 of the cDNA, we discovered, as expected, that the controls have one single band representing the longest splice isoform. We found that the mother and the father had both the long splice isoform and a further, which was missing approximately 87 base pairs. And as expected, the patient had only the shorter splice isoform, missing the 87 base pairs. And in the patient's cDNA, there was no full-length isoform present. So this gave strength to our hypothesis about a potential hypomorphic allele, which was leaving a residual partial function of the TRP11 protein. However, what we didn't expect was the presence of a further larger band here in the patient's cDNA, which was also very faintly present in both the mother and the father. So we did some sequencing of this PCR product, and we discovered that, as well as the presence of the normal splice isoform and the splice isoform missing exon 9, we also had a third cDNA product, which included approximately 35 nucleotides at the beginning of intron 9 of the TRP11 gene. And so we did some analysis using a splice analysis algorithm, and we found that there was a predicted cryptic splice site at position exon 9 plus 33 nucleotides, which was expected to cut exactly after 35 base pairs. So the prediction is that the mutation is causing both the deletion of exon 9, plus it is also causing, due to the lack of the normal splice site here, it's causing activation of a weaker cryptic splice site further into intron 9, which causes the presence of this slightly longer splice isoform. Now, we haven't yet been able to do protein studies, but if this protein is expressed, it may also be participating in the occurrence of the phenotype in these patients. You would expect, because this mutation causes a protein truncation after 20 amino acids, after the end of exon 9, you would expect that this would result in nonsense-mediated decay. However, the fact that the exon 9 plus 35 nucleotide splice isoform is detected in the patient's cDNA indicates that an abnormal protein may also be present. So we're currently working to isolate protein from the patient's cells so that we can do Western blotting to demonstrate the presence of the protein. And if this longer and if this truncated splice isoform is present, then we will go ahead and do some functional cellular studies using potentially stem cells that we can differentiate into chondrocytes. So the conclusion is that the in-frame splicing mutation produces a hypomorphic allele, and also that the largest frameshift allele, if translated, may contribute to the phenotype. As I said, as we were preparing our manuscript, a group in Europe published the TRP11 gene as the cause of odontochondral dysplasia, but shortly afterwards we also published our paper, which is cited here. Now, the second skeletal disease that I want to talk to you about is called multiple exhaust disease. And this is a skeletal phenotype which is associated with the growth of benign tumors at multiple sites, especially at the epiphyses of the long bones of patients. In the majority of cases, they don't progress to malignant tumors. However, as you can imagine, due to their proximity to joints, they cause significant discomfort and even pain in the patients. And many of the patients undergo multiple surgeries to try to remove these exostocies. Now, multiple exostocies are caused by loss of function mutations, autosomal dominant loss of functional mutations in the EXT1 or EXT2 genes. The EXT1 and EXT2 genes are exostosin 1 and exostosin 2. They're predicted tumor suppressor proteins, and they are highly expressed in cartilage as well as in other tissues. Their functions, they function within the endoplasmic reticulum, where they have a role in the production of glucosaminoglycans for the production of sulfated glycoproteins, which are a large part of the extracellular matrix of cartilage. And so what seems to be happening is that when you have loss of function mutations in one or other of these genes, the hetero-oligomeric complex that is formed between the EXT1 and EXT2 proteins loses at least part of its function, and so we lose some of the integrity of the extracellular matrix. Now, this causes a disorientation of the chondrocytes in the cartilage of these patients, and also deregulates the chondrocyte proliferation, causing these benign tumors. So we analyzed a large family with multiple hereditary exostases, and the family is indicated here. It's obviously autosomal dominant, and all of the people who were marked with asterisks here were people that we analyzed in the DNA looking for mutations. So in this case, we used a cancer panel, which was sequenced on the Ion Torrent platform within our laboratory, and we found a mutation in the EXT1 gene, which, as is the majority of cases, was a predicted loss of function, being a nonsense mutation after 97 codons of the EXT1 gene. Now, being a nonsense mutation, again, we started to think about nonsense-mediated decay. And so once again, we took peripheral blood cells from some of the patients, we synthesized cDNA, and we looked to detect the EST1 transcript. Now, nonsense-mediated decay, for those who are not aware, is a situation in which when there are nonsense mutations in a gene, the cell seems to have a mechanism whereby, rather than wasting energy expressing the gene and translating the protein, it is able to destroy the transcript by detecting the presence of the nonsense mutation. This only occurs if the mutation is present before the last exon of the gene, which is the case in our situation. So we isolated cDNA and we searched for the EXT1 transcript, and the result is indicated here in this part of the graph, where in our proband patient and her mother, we have a reduction in the level of the EXT1 protein. The EXT1 transcript. As expected in the patient, we have almost exactly a 50% reduction, and this reflects the fact that we have an autosomal dominant disease. So in other words, we have one allele which is producing normal transcript and normal protein, and the other allele which is null. And so this should reflect approximately a 50% reduction. In the mother's case, there was a slightly higher level of expression, but still it falls within the range of expression level that could be attributed to nonsense-mediated decay. Now, we started to think about further investigations into multiple hereditary exostases, and recently there was published a paper which suggested that when you have null mutations in a given gene within a patient, that we can see a situation where related genes might actually increase their expression. And so we start as pretty much as a compensatory method. And so we started to think about the possibility that EXT2, the related gene, might increase its expression in patients which have mutations in the EXT1 gene. And so again, we did analysis of the EXT2 transcript in the same proband and her mother, and we found that in the mother, there seems to be indication of an increased expression of EXT2. However, this wasn't reflected in the proband. So we're currently doing new cell isolations where we're going to repeat this experiment to see if we can resolve whether we really have a compensatory upregulation of EXT2 or not. Now, the third skeletal disease that I want to talk to you about is called Fatco Syndrome. And in this case, we haven't yet identified the gene. It's a rare skeletal disorder. Fatco stands for fibular aplasia. And you can see here in this patient, for example, the fibula is missing. Tibial campomelia, and this is bowing of the tibia here in the lower limb, and also oligosyndactyly, which we can see here both in the photos and in the x-rays. Oligosyndactyly suggests that you have missing bones in the phalanges, and also that you have fusions. So we can see here that we have missing phalanges, and we also have fusions in the hands and also in the feet. Now, Fatco syndrome is a rare syndrome. The gene is unknown, and there are very few cases reported in the literature. The last time we looked, there were approximately 17 cases reported in the literature. It has clinical overlap with another syndrome called Fuhrmann syndrome, which is very similar. And for this, the gene is known. It's caused by mutations in the Wnt7a gene, which is part, obviously, of the Wnt pathway. Now, due to the fact that there is such clinical overlap, for the patients that we encounter, the first step that we undertake is sequencing of the Wnt7a gene. Now, despite the fact that Fatco syndrome is very rare worldwide, we're lucky enough here to have a cohort of 7 cases. 2 of these we have identified from Brasilia. There have been 2 that have been identified in Rio Grande do Sul, 1 in Ceará, and we also have managed to collect 1 patient from Chile and another who was sent to us from the United States. So relatively speaking, we have quite an impressive cohort. All of the cases that we have the following inheritance pattern, where the parents are phenotypically normal and the child is affected. Now, this could suggest either new mutations, or it could suggest autosomal recessive inheritance, among other possibilities. So in order to analyze these patients, the first thing that we did, as I already mentioned, was sequencing of the Wnt7a gene, and all of the patients were negative for mutations in the gene, suggesting that we didn't have any cases of Fuhrmann syndrome. The second thing we did was to think about the possibility of chromosomal anomalies. Now, our previous speaker, Professor Aline, mentioned array analysis of chromosomes, and we did exactly this. We did chromosomal microarray analysis, and again, none of the patients showed any obvious chromosomal rearrangement, deletion, or duplication, which might have been associated with this syndrome. So the next thing we started to think about was exome sequencing, and exome sequencing is a method whereby it's based on the knowledge that 85% of all mutations causing monogenic diseases are located within the coding regions of genes. Now, due to the fact that if you join together all the coding regions of all the genes in the human genome, it amounts to only 2% of the genome. This means that you have an enormous cost-benefit ratio. You have 85% of mutations, which are found in approximately 2% of the genome. And so we decided to take 5 of these trios, and we did complete exome sequencing in them, and then we analyzed the data using different parameters. First of all, we used a hypothesis tree analysis, which means that we didn't give any regard to potential inheritance methods, inheritance patterns. We were simply looking for mutations in potentially skeletally related genes, and also we were working on the hypothesis that at least 2 of the patients should share mutations within the same gene. We also looked at candidate genes, focusing in genes that are already associated with skeletal diseases. We discussed some possible inheritance patterns. Because the inheritance pattern is unknown, there have been suggestions of X-linked inheritance, of autosomal dominant with new mutations, of autosomal recessive inheritance, and so we considered all of these possibilities. We also looked for de novo variants, suggesting that these variants are not present in the parent, but only in the patients. And we also, of course, brought into account allele frequency, looking for super rare variants, and we used a number of predictive algorithms. Now, combining all of our 5 trios, we found a total of 490,000, a little more than 490,000 variants. We found 83 variants in candidate genes. However, those that had a strong predicted impact on the protein were only 4. And finally, when we took into account allele frequency parameters, there were only 2 variants. And this is actually a very good result when you talk about exome sequencing. What you really want to do is filter a large number of variants down to very few. However, none of these 2 variants was present in more than one of our patients, which decreased our confidence in any one of them. And also, even though they did fall into candidate genes, neither of the genes that was involved was a really strong candidate for this type of phenotype. And so after sequencing 5 trios for the complete exome, we were still none the wiser as to the potential genetic variant that might be causing Fatco syndrome. And so our current strategy is based on the increasingly convenient availability of whole genome sequencing. And so we currently have 5 patients who we have sent to Sao Paulo, and we are sequencing the entire genome on the Illumina platform. Now, as well as this, we have also been lucky enough to isolate cells, again, blood cells, from 2 of our patients. And so we are also in parallel initiating functional studies in these patients. And our initial focus is going to be on transcriptome sequencing and also on analysis focused on the Wnt pathway. Now, even though we're sequencing the whole genome, we are aware of the possibility that we still might not identify the genetic lesion. And again, Professor Aline talked to us about epigenetic variants. It is possible that this syndrome might be caused by some disturbance in the epigenetic profiles of these patients. So after analyzing our whole genome sequencing, which is going to take some time, if we still don't identify the genetic lesion common between the 5 different patients, we're going to have to think about the possibility of doing epigenetic analysis for these cases. Now, one of the challenges that we have in analyzing skeletal diseases is that, first of all, it's really difficult to access the cells that are involved in skeletal diseases, which are the chondrocytes. It's very invasive to take out chondrocytes from a patient, and so ethical approval would be very unlikely. Secondly, most of the events involved in the development of these diseases, which are usually patterning events, they occur during embryogenesis. So obviously, we really can't get embryonic chondrocytes from these patients. So what we've been thinking about is methods of using non-chondrocytic cells from our patients, for example, the blood cells that I've been talking to you about that we've been able to isolate, and using these to study skeletal development. Now, one method by which you could do this would be to take the blood cells and induce the formation of induced pluripotent stem cells. However, we've been looking at other methods. We've been looking at using extracellular vesicles for the induction of chondrogenesis, and we've also been looking at methods of transdifferentiation, which means that you go straight from one somatic cell, for example, a fibroblast, and you convert this into a chondrocyte. Now, I'm not going to say anything more about transdifferentiation because our work in this regard is very preliminary and I don't have very interesting data to show you, but I would like to talk to you a little bit more in these last few minutes about extracellular vesicles and the potential that they have to induce chondrogenesis. Now, extracellular vesicles are vesicles that are secreted by all cell types. There are different categories. There are microvesicles, which are produced by budding, as we see here in this figure, and also, which is much more interesting for many research groups, are exosomes, which are produced initially in intracellular multivesicular vesicles, and then by fusion of these vesicles with the cytoplasmic membrane, these exosomes are released to the extracellular environment. These different types of vesicles can be categorized based on shape and size and also on content. The microvesicles in general, extracellular microvesicles, they have a size between 150 up to about 1,000 nanometers in diameter, whereas the exosomes have a diameter more in the region of 40 to 100 nanometers. Now, the roles of these vesicles are diverse, and they're also not completely understood so far, but they are thought to have a strong role in intercellular signaling. Studies for characterization of these vesicles is currently intense, and it has been shown that they contain, as well as lipids, many different proteins, and they also contain RNA and DNA. Of all the RNA species that have been researched so far, all of them have currently been detected within exosomes. So you can find messenger RNA, microRNAs, long non-coding RNAs, circular RNAs, and even you can find transport RNAs, transfer RNAs. So what we, thinking about these extracellular vesicles, we wondered whether vesicles derived from cultured chondrocytes could induce chondrogenesis in stem cells. So what we did was we took chondrocytes from newborn mice, we isolated them, and we cultured them in the laboratory. Now, with these chondrocytes, we collected the conditioned media, and we isolated the extracellular vesicles. There are several different methods for isolating these vesicles, and none of them is perfect, and it's quite debated in the literature. But the method that we used was the most common, which is a series of centrifugation steps culminating in ultracentrifugation at 110,000 G. So when we did a profile of these isolated vesicles using a piece of equipment called a Q Nano, we discovered that we had a good concentration of vesicles, up to 10 to the power of 10 per mil, and that they fell within the expected size range, and that the majority of the vesicles were actually in the range that would be expected for exosomes, as well as some larger vesicles as well. When you work with extracellular vesicles, and especially exosomes, it's common to demonstrate that you really do have exosomes. And so one of the most common methods is to do Western blotting for markers of these vesicles. And I apologize that these blots aren't so strong, but we do show the presence of the known extracellular vesicle markers, exosome markers, including TGS101, CD9, and CD63. As well as this, although I don't have an image here, we also did electron microscopy to show that we did have extracellular vesicles in the expected range of the exosome bodies. So what we did then was we took these vesicles and we applied them to initially mesenchymal stem cells and then to induced pluripotent stem cells. And so what we have here, first in this panel, is a series of samples where we have negative control, positive control, which induces mesenchymal stem cells to undergo chondrogenesis. This is a commercial chondrogenic media produced by Thermo Fisher. We also used conditioned media, which is well described in the literature as a method of inducing chondrogenesis. We further used conditioned media from which we had removed the extracellular vesicles. And finally, we also applied the extracellular vesicles themselves. Now, although we didn't have a gene expression quite as high as in the positive control, we were able to show that the extracellular vesicles alone can induce expression of SOX9, which is the master regulator of chondrogenesis, and also induces expression of type 2 collagen and aggrecan, which are strong markers of chondrogenic differentiation and the chondrocytic phenotype in general. We also did Alcyon blue staining of the extracellular matrix. Now, this is only after 7 days, and so the staining isn't so strong. Usually we would go to at least 4 weeks of differentiation, but what we can see here is that the negative control doesn't show any staining, whereas the positive control and also the cells that have been treated with the extracellular vesicles do stain with blue. Now, Alcyon blue is a chemical that stains sulfated proteoglycans in the extracellular matrix of cartilage. So it is an accepted method of demonstrating that you have chondrogenic differentiation. We also applied these vesicles to induced pluripotent stem cells. And you may already know, but let me briefly just tell you that induced pluripotent stem cells are cells that have been terminally differentiated, for example, fibroblasts. And Yamanaka and colleagues in 2008 showed that you can induce forced expression of a surprisingly small number of genes, and that you can cause terminally differentiated cells to revert to a pluripotent phenotype where they are very similar to embryonic stem cells. So we got these IPS cells, induced pluripotent stem cells, and we applied the extracellular vesicles to these also. And so here we have the cells at T0, also after 7, 14, 21, and 28 days of stimulation. And what you can see here after 28 days is that we do have some quite strong foci of Alcyon blue staining. And this is quite typical of IPS cells. They tend to grow in clumped colonies. And also we found that with the progression of time after 0, 7, 14, 21, and 28 days, we start to see quite strong expression of SOX9, the master regulator of chondrogenesis, and also of type 2 collagen, which usually accompanies and usually follows SOX9 expression. So these data suggest that we can use chondrocyte-derived extracellular vesicles to induce chondrogenesis of stem cells. And so we're thinking that this is going to be quite a useful method, because it means that we'll be able to take cells from our patients, which may be stem cells or maybe somatic cells that we can induce pluripotency, and then we can use chondrocyte-derived vesicles to induce chondrogenesis in these cells. And this means that we will have a tool to study the whole process of chondrogenesis from pluripotency to terminal differentiation and try to discover what is going wrong in these different diseases. And so this is a process that we already have underway in the case of Fatco syndrome. And so our idea is that even if we don't identify the causative gene or the causative genetic lesion, we will be able to already initiate our functional studies and try to get some insight into the molecular pathways that are going wrong in the Fatco syndrome. Now, as well as all this, we also showed that the extracellular vesicles are associating with the cells. So here we have our mesenchymal stem cells, which are shown here using the DAPI stain, which stains the nucleus of cells. And we also have the vesicles which have been applied in, and they're stained with the green stain here. And so this image here, it's perhaps not too strong, but it does show the vesicles associating with the cells. But we also used a software to show that we do have considerable overlap between the nuclear stain and the extracellular vesicle stain. Now, we also did some protein analysis of the vesicles, and we found that the vesicles, among the proteins that they contain, they do contain the SOX9 protein. And this was to be expected because SOX9 is a master regulator of chondrogenesis, and so it suggests that chondrocytes can produce vesicles which contain this protein, which alone is sufficient to induce chondrogenesis in stem cells. We also did some gene expression analysis in the extracellular vesicles, and we showed that they did contain transcripts for many of the cartilage-associated genes, including type 2 collagen, aggrecan, SOX9 again, and some of the FGFR proteins, which are the receptors for the fibroblast growth factors very associated with chondrogenic development. It also contains the WDR5 transcript, which is another transcript that is known to be associated with the control of chondrocyte proliferation. Importantly, these vesicles do not contain either the type 10 collagen or the Indian hedgehog transcripts. And these are transcripts that are more associated with terminal differentiation of chondrocytes when they begin to enter into the hypertrophic stage of their development, and they eventually die. And this is the point at which we get substitution of cartilage by bone. And so if we're thinking about chondrogenic development and induction of chondrogenesis, we don't want to see the presence of these transcripts because they could really be a negative influence on the type of chondrocytic phenotype that we are trying to develop. So currently we are investigating the capacity of the extracellular vesicles to inhibit chondrocyte dedifferentiation. Sorry, this should say dedifferentiation here, because dedifferentiation is a problem when you culture chondrocytes in vitro. After some time, they tend to lose their chondrocytic phenotype, and we do already have some preliminary data suggesting that our vesicles can inhibit the dedifferentiation of these chondrocytes. We're also analyzing sequencing data. We're also analyzing sequencing data for RNA and DNA, which we produced by the nanopore platform. And so last year, we took some of these vesicle samples to Ireland, where we did nanopore sequencing to analyze the transcriptome and also the DNA profile in these chondrogenic extracellular vesicles. So in summary, we are analyzing a range of different skeletal phenotypes, and currently we are also looking at new methods of inducing chondrogenesis so that we can improve our capacity to do functional studies in our patients. So thank you for your attention. I should acknowledge some people here who aren't just the people who did the work, but really are co-authors in this work. Christina Medina is a PhD student who is working on the Fatco syndrome and also did the work on odontochondral dysplasia. Felipe Marquez does a lot of our work on exome sequencing and is also involved in the whole genome sequencing. And Lana Aguiar was the person who did most of the work on the chondrogenic extracellular vesicles. Thank you.
Thank you, Professor Robert, for your excellent lecture. A lot of information, very interesting subject. I learned a lot. It's not my area of expertise, so I pay a lot of attention and learned a lot. Thank you so much. I'm afraid we are running out of time for the audience questions. So once again, I would like to thank you for accepting our invitations.
Thank you.
And for being here with us. It's always a pleasure. Thank you so much, Professor. And now...
I just like to say, okay, may I? Sorry. I just like to thank you very much, Robert, for accepting this invitation to have time to talk to you backstage. So thank you. You are such a nice friend and a great researcher. And if it's possible, I'd just like to make one question.
Of course.
May I? Robert, I'd like to know if you have seen some cerebrocostal mandibular syndrome.
I have no. We don't have any patients for that syndrome. I haven't come across it.
You have one patient in where it's stuck in the analysis because the exome sequence show a mutation, the gene that is probably the gene that causes the syndrome. But you have there's some MD that do not agree if this is the right syndrome. So maybe we should talk.
Yes,
you can give some opinion about that. Okay.
Yes, always a pleasure.
Okay. Thank you very much, Robert.
Thank you, Professor. Now I'm going to introduce you to our next speaker, which is Gisele Almeida Amaral. She has a degree in pharmacy and biochemistry from the Unified Institute of Higher Education, Objective. She is a specialist in clinical pharmacy from the Catholic University of Goiás. She has a master's degree in basic and applied immunology and parasitology at Federal University of Mato Grosso. And now she is a PhD student of the Biological Science program at Federal University of Goiás. Welcome, Gisele. Thank you for being here with us.
It's my pleasure.
It's up to you.
Well,
I can hear you.
Okay.
Now you can share your screen and start your presentation. Feel free. It's up to you.
So, good afternoon. My name is Gisele Almeida Amaral Fonseca, doctoral student at the graduate program in the biological science, and my advisor is Dr. Fernanda Vitorino, and I'm here to present part of my doctoral work. The adrenal gland's cortex secretes a group of hormones called corticosteroids, and according to their chemical structure, are classified into mineralocorticoids, androgens, and glucocorticoids. Mineralocorticoids act primarily in electrolyte transportation and water distribution in the tissue. Androgens act on secondary sexual characteristics in specific organs, while glucocorticoids exert an important effect in elevating blood glucose concentration, as well as it acts in the metabolism of lipids and proteins. So the action of the glucocorticoids is directly related to the modulation of gene transcription. Due to their lipophilic nature, glucocorticoids cross the cytoplasmic membrane of the target cell by passive diffusion. Once linked to their specific receptor, called glucocorticoid receptors, they enter the cell nucleus and bind to promoter regions, called glucocorticoid responsive elements through mechanisms called transactivation, inducing synthesis of anti-inflammatories such as lipocortin 1, nuclear factor Kb inhibitor, and systemic metabolic proteins. Glucocorticoids also act by a genomic mechanism Glucocorticoid receptor complex binds to a coactivate molecule with high stone acetyltransferase activity, leading to inhibition of pro-inflammatory nuclear transcriptor factors such as NFKB. This process is called transrepression and leads to decreased expression of cytokines and pro-inflammatory factors. A commonly used glucocorticoid is prednisolone. It has good oral biodisponibility and wide therapeutic application. It can be used in pathologies such as endocrine disease, muscular pain, dermatological disorders, and allergic and respiratory reactions. Also, in some hematological diseases, the problem is that most of the treatment, the problem with this glucocorticoid is that most of this treatment, the patients has to take this drug for a long period of time, and it can lead to appearance of many side effects like induction of diabetes, myopathies, physiological changes, and in special, artery hypertension. During some time, it was believed that hypertension was only caused by an enhancement of transepithelial sodium transport because of dysfunctions on the 11 beta hydroxy uh, 11 beta hydroxysteroid dehydrogenase enzyme. And this enzyme can come in two important isoforms. For activation, we have the 11 beta HSD1, and for inactivation, we have the 11 beta HSD2. Both are present in the renal tissue. So when the concentration or when the glucocorticoids are in excess in this organism, they have an agonist action on mineralocorticoids receptors, and the excess of it leads to an increase of 11 beta HSD1 or uh, decrease on the 11 beta HSD2, which acts in some in some mineralocorticoid receptor in a secondary or non-specific way. So it was believed that a large part of the pro-hypertensive action of this drug was exclusively due to this renal mechanism. So of increased of sodium retention and also increase of potassium excretion, and consequently water, and generating a model of hypervolemic hypertension. The filtration process in the kidney depends on the amount of blood that goes through the renal artery and it's filtered in the glomeruli. So, an adequate control of vascular function and renal perfusion is necessary for the renal filtration process to occur satisfactorily. Dexamethasone, a glucocorticoid with high anti-inflammatory potency, has demonstrated hypertensive effects for some time. In this work, dexamethasone induced alterations in the level of proteases involved in blood pressure homeostasis and blood coagulation in rats. Dexamethasone Dexamethasone also has been widely used in an experimental model for inducing arterial hypertension. In this work, it is possible to verify significant differences in blood pressure levels in animals treated with dexamethasone, blue line, while when compared to control pink line. However, studies studies with prednisone in this regard are not widespread. Kidney plays an essential role in the regulation of blood pressure through self-regulation, as shown. Some evidences indicate that the use of glucocorticoid negatively influences this physiological feedback. There are few studies that demonstrate how prednisone influences the vascular of the renal artery. So, in addition to assessing whether blood pressure is reaching the renal artery, another point is to verify which type of injury this model of prednisone could generate in the kidney. Uh, so, the hypothesis of our work is that prednisone increases blood pressure, leading to a vascular dysfunction, tissue damage, and renal dysfunction. So, the aim of this study was to analyze the renal artery vascular response, as well as the renal morphology and function after the administration of prednisolone in oyster rats. So, male oyster rats, 280 and 320 grams, were maintained in the facility room with 12-hour light dark circle, fed in a standard shown diet, and free water intake at the temperature of 24 Celsius degrees. From now on, our control group will be called CTR and prednisone group PRD. Each group had 8 animals, and the control group were treated with vehicle called Cirspend, adjusted volume to body weight, or prednisone 5 milligrams kilogram body weight by gavage once a day for 17 days. For these experimentals, we followed the procedures. We analyzed the blood pressure by tail plethysmography, renal function by the dosage of creatinine, urea, and microalbuminuria, and renal morphometry with histological analysis. The kidney tissue were stained with hematoxylin and eosin, periodic acid sheath, and serous red staining for evaluation of morphological damage. Statistical analyses were performed by unpaired student t-test or one-way ANOVA followed by Tukey post-hoc test. Values of p less than 0.05 were considered statistically significant. So, the first question was, does prednisone treatment affects blood pressure and kidney function? As you can see, the table shows that there was an increase in systolic blood pressure in the comparison between groups on days 0 and 17. In relation to the parameters of feed and water consumption, there was no difference between the group, as well as the urinary flow and serum of urinary creatinine and urea dosage. However, when we analyze microalbuminuria dosage, it is possible to observe an increase in the prednisone group. The term microalbuminuria implies a relatively small amount of protein in the urine, and therefore it is used as a powerful predictor of kidney disease and mortality in individuals. Uh, and does prednisone affect renal morphology? When we analyzed the way of the kidney, it didn't show any difference, neither in the cortical or medullary area of the kidney, as you can see in the blue zone. When we analyze the glomerular capsule, no statistical differences were observed when comparing the groups. However, it is possible to observe a decrease in the internal glomerular area of rats treated with prednisone. Fernandez reports that type of glomeruli present in hypertensive nephrosclerosis is the ischemic glomerulus. We also observed that the prednisone group presented an extracellular matrix deposition in the renal tubules region, showing a greater amount of amorphous fundamental substances, glucosamine glycans, acquiring this pinker color by periodic acids when we compare prednisone group and control group. In a study carried out by you, hypertensive animals by DOCA salt model also had a greater deposition of extra matrix in the region of the glomerular capsule, giving rise to glomerulus sclerosis. It was also possible to observe or to verify that the animals treated with prednisone presented an increase in the deposition of collagen fibers in all its extension, which is characterized by the more reddish tone of this tissue. During tissue analysis, I began to observe that the appearance of prednisone vessels had a greater deposition of collagen fibers. So, after quantifying this perivascular collagen, it was possible to confirm its increased. Type 1 fibers appear as thick fibers, highlighted, very refringent, and red in color, while type 3 fibers are presented in thin benders with thick fire with a weak refringence and a greenish yellow color. A greater deposition of type 1 collagen in a greater deposition of type 1 collagen in vessels of animals treated with prednisone demonstrated renal perivascular remodeling, since this type of collagen is synthesized by fibroblasts and vascular smooth muscle cells, justifying the increasing in microalbuminuria in these animals and one set of kidney injury. So the next step of our work was to evaluate the expression of alpha-action isoform in smooth muscles, and it was possible to verify that prednisone presents an increase of the same. The expression of this protein by vascular smooth muscle cells and by fibroblastic cells of the stroma is associated with pathological condition that leads to fibrosis. Alpha smooth muscle actin is the actin isoform that predominates within smooth muscle cells and plays an important role in the fibrogenesis. So, in our final disclosures, we observed that normotensive rats treated with 5 milligrams kilogram by 70 days with prednisone presented arguments blood pressure, renal morphology, and functional alterations. I'd like to thank the organization of the symposium for the opportunity. I'd like to thank you, Professor Paulo Guedini, for having received me so well in his lab. And I'd really like to give a, a very special acknowledgement for Professor Manuel and Professor Fernanda for the help in the histological experiments and all my co-workers in UFMT. And of course, I'd like to thanks my advisor, Fernanda, for this amazing help. Thank you very much.
Thank you again, Gisele. Excellent lecture. Thank you so much. I think we have a little bit of time for the audience questions. I think we have a question here from pós-graduação em Ciências Biológicas. I think it's Professor Carlos. Gisele, thank you for the very nice presentation. Did you analyze the heart histology in prednisone-treated rats?
No, Professor. Unfortunately, we didn't think about it, but it's a nice suggestion.
Okay. I think we have another from Elizabeth Pereira Mendes. Congratulations of your presentations. Have you noticed if there are changes in tubular frequency or morphology?
What I didn't understand what she, Elizabeth, what do you mean about tubular frequency?
Can
Changes
you,
in the tissue?
Elizabeth, can you write in the chat, please?
If there are changes. Well, as I showed in my presentation, when we analyze the proximal tubules and the distal tubules, you can see this deposition of collagen, and apparently we can see more. We could see diminish and the capsule, the glomerular capsule, and so that's one of the reasons that it leads us to think that the microalbuminuria has increased.
Okay, she complements her questions and said number. I think it makes sense now.
The number of the tubes.
I think so.
No, we didn't measure the number of the tubes.
Okay. Professor Lucilene, do you have any questions to complement?
No questions, just to congrats Gisele for the brilliant presentation. Very nice work.
Thank you very much.
Now I don't have questions. I'm trying to be very quiet now, okay?
Okay, feel free to ask anything. I think you said a little bit in the final of your presentation, but I like to know what is your next steps. How long have you complete your PhD? Do you have any more time?
I haven't brought to this presentation because of the time, but we did all the vascular analysis of the renal artery, and I have all these results. And now I'm just writing my work, right, to submit, and it's done. We're just finishing to write, to submit the paper, and then it's finished. Hopefully it will be accepted soon.
It will be.
I'll just
fingers crossed.
Yes, I'll just present my homework soon.
Okay, it's nice. Thank you. Thank you so much. I think Elizabeth said Another thing, because cortisol could be changes the vascular of tubule area.
Okay. Thank you very much, Elizabeth, for your
Okay,
collaboration.
thank you again, Gisele, for your presentation, sharing with us your results.
Thank you.
It was a pleasure having you here. And now we are going to have a break of less than 30 minutes, and we're gonna return to our next session at 4:15. Feel free to use the bathroom, drink a little water, and see you in our last session. Bye-bye.